Let's talk about meiosis. Unlike mitosis, which is a similar process, meiosis only happens in specialized cells, the sex cells, or germ cells. This would be in the gonads in people. Plants, it would happen in ovules or the anthers to make pollen. This cell has probably already done mitosis already, and so it's ready to go and make gametes. In humans, that would be sperm or eggs. And so I'm going to assume that we've already gone through interphase, in which replication occurred and made sister chromatids for me. So I'm going to jump right into the middle of meiosis. Not the middle, sorry. I'm going to jump right into meiosis and start with prophase. Now, since meiosis happens in two sets of divisions, I'm going to call this prophase 1. And it's going to be really similar to the mitosis prophase. These chromosomes would have condensed. I'm just not showing that step. But something else fairly important happens besides the typical things that we would associate with prophase, like the nuclear envelope going away. And now I'm going to move together. Microtubules would be doing this in your cells. I'm going to move together the homologous chromosomes. Those are the ones that are similar. They carry the same type of genetic information. Long ones with long ones, medium ones with medium, and short with short. In the humans, we have 23 pairs of these. Here I'm just showing 3 pairs for simplicity. So we have homologous chromosomes. So at this point, we're crossing over. Some swapping of DNA can occur. And that happens during prophase 1. All right, so as those microtubules start moving these homologous pairs, or a tetrad, so there's 4 strands of DNA you'll notice in each of these, so those are tetrads. They're being moved to the middle, and once I get to that middle phase, now I'm in metaphase 1. Looks a lot different than metaphase of mitosis because we have these lined up 2 by 2. They don't have to line up this way. They could have lined up with the red on one side and the blue on another, or like this, or any combination of those. That becomes important in genetic shuffling. This is called independent assortment. The next phase, just like during mitosis, is anaphase. This will be anaphase 1, where those homologous pairs are pulled apart. And as we get into telophase 1, then our nuclear envelopes come back, cytokinesis happens, and we get a new cell. Now, we're not entirely done. We have to start over at the beginning because I don't have these sister chromatid pairs split up. And so I'm going to go back to prophase again. This time this will be prophase 2, which makes sense. And here I'm going to get rid of the nuclear envelope. I know it seems wasteful, I just put it there, but now I'm going to get rid of it. I'm going to start moving these guys to line up, and that will be our metaphase 2. Same thing over here. Just going to line them up this direction. And in anaphase 2, they get pulled apart by microtubules. Now the sister chromatids have split up. I have them organized already. Telophase 2, along with cytokinesis, is going to divide these up into 4 different cells. We're going to need, I won't do it for all of them, a nuclear envelope around each. And you'll notice that even though we started with 6 chromosomes in the original cell, we now have gone to 3. That's half of the chromosome number. These are now a 1N or haploid cell. In humans, that would be a sperm or an egg cell. Half the amount of chromosomes ready to fertilize the other gamete and create a diploid zygote. So there's an overview of meiosis. We get 2 rounds of division, similar to mitosis, but with 2 rounds of division, and we end up with half the genetic material in each cell.